My name is Gene Bowman. I am the clinical trials director at the McCance Center and PI of the Brain Nutrition Lab. I moderate these sessions, and thank you all for attending. Before we get started, there's a couple of housekeeping changes. In the future, you will have an invite for this seminar series coming from the MGH McCance Center for Brain Health, an Outlook calendar. Okay, as opposed to our program manager here at Shrestha's calendar. Also, our next seminar will be taking place on a Friday as opposed to a Monday. We've trying to accommodate other speakers by providing a Friday slot for those that are booked on Mondays or have other obligations. So the next one will be Friday, May 30th. And we will post who the speaker is there. It's Dr. Britton on causal inference in dementia prevention research. Can national public health policies reduce the risk of dementia? Um, so, Dr. Tansey, would you like me to introduce the speaker today?
Yes, thank you.
Okay. All right. So, we're lucky to have Dr. William Eimer here, who's an instructor in the laboratory of Dr. Tansey in the Genetics and Aging Research Unit, and also an affiliate of the McCance Center for Brain Health and Neurology Department here at MGH and Harvard Medical School. Dr. Eimer dual majored in biology and psychology at Carnegie Mellon University before receiving his PhD in neuroscience from Northwestern University under the guidance of Dr. Robert Vassar. He continued in the field of Alzheimer's, working as a postdoc under Robert Moore and then as junior faculty member of the Genetics and Aging Research Unit here at MGH. And he's continued and expanded the exploration of amyloid and tau as host defense proteins, a path started by the late Dr. Moore. He aims to decipher better the connection between the pathogens, immune response, and Alzheimer's disease, leading to more effective treatments. The title of his presentation today is Amyloid Beta and Tau, an essential link between the immune response and Alzheimer's disease pathology. Welcome, Dr. Eimer.
Thank you. Let me bring up my presentation. All right. So thank you for the introduction. Today, I want to discuss some of the work that we've been doing over the last 4 years since I gave a McCance seminar and how that has impacted our outlook and our movement forward in studying this connection between amyloid beta, tau, the immune system, and Alzheimer's disease. So start with a classic figure that most of you have seen. This shows the increase in different molecular markers as we progress from normal all the way to dementia and Alzheimer's disease. And the general premise of what this has created in the field is this concept that we start with amyloid, we progress to tau tangles, and then this lands in neuroinflammation, creating the dementia that we see in AD. And almost a quarter century ago, this amyloid cascade hypothesis was established, and this has been the dominant hypothesis of how AD functions. We get the amyloid beta, specifically the 42 isoform. It starts to oligomerize. It impacts the microglia and astrocytes, the synapses. We see neuronal death at some point in this process, possibly earlier, possibly a little later. We see this hyperphosphorylation of tau, development into tangles, and then the neuronal death and dementia. This flow has been what research and pharmaceuticals have chased over the last 25 years in trying to find an effective treatment to dementia. And while we have had some mild success with drugs recently, it hasn't been a silver bullet. We haven't found something that absolutely cures or a definitive pathway from a normal, healthy human being to developing Alzheimer's disease outside of familial Alzheimer's disease, where we know the genetic mutations and the overexpression of amyloid that develops there. But that's 5% of what we see in Alzheimer's. What about the sporadic? And a new hypothesis, well, a new old hypothesis, has been the involvement of pathogens in this process. Uh, since the discovery of Alzheimer's disease in 1909, even the first examination of the brains, it was mentioned that it shared similarities to other known infections in the brain. And in about the time that the amyloid cascade hypothesis was established, there was initial evidence about the presence of HSV1, herpes simplex virus, potentially having a role in AD. And as other diseases like multiple sclerosis have found a involvement of pathogens, the idea of a microbial involvement of in Alzheimer's disease has become more attractive. But we know definitively that amyloid beta and tau are part of this disease. So how do we find, what is that interconnection that leads from pathogens to Alzheimer's involving amyloid and tau? And this is where antimicrobial peptides come in. Dr. Rob Moore and Rudy Tanzi, 15 years ago, sat down and realized that not only were there some innate immune genes that seemed to be risk factors for the development of AD, but the actual structure of antimicrobial peptides was very similar to amyloid. We had 12 to 15 amino acids long, charged, and this development of alpha helix and beta sheet conformations in the secondary structure. And as you can see on the right here, we're looking at LL37 and beta defensin. They form these nets and aggregates around the targets that they hook into. So this idea that maybe amyloid wasn't just hanging around without a purpose, but maybe it actually was involved in the innate immune system. And lo and behold, the first paper that came out in 2010, that's what that connection was. They found that amyloid beta was effective against certain strains of bacteria that were tested. And we developed, Rob and then Deepak and then myself, each kind of contributing successively to this model that amyloid beta would bind to the outside of the pathogen. It could isolate it from the host cells, preventing the infection process and the damage to the host cells, and then ultimately agglutinate these pathogens together as a mechanism for clearing them out. My particular work when I joined the unit was to take this idea and apply it to viruses. And we looked at HSV1 and HHV6 as two rather penetrant viruses in the community and ones that were known to make it into the brain. And what we found was that amyloid was extremely successful at attacking these viruses, binding them and sticking them together. And it also worked on HHV6, although not quite as aggressive as we saw with amyloid. When we took this to in vitro models with the 3D culture and the mice, we were able to see an acceleration of the pathology in the 3D culture that would usually take weeks to develop, we could do in a day. And the mice also showed this clumping of HSV1 into plaques in the brain that looked phenomenally similar to those that we would see in humans. And on the right, what you can see is if we take away all the cells and we just have a 3D matrix with amyloid and virus in it, that is sufficient to form what is a plaque, and a rather large plaque at that. You can see the green is the amyloid and the red is the HSV1. And the amyloid was able to aggregate and pull the viral particles together into this pseudo plaque. So, along with the work that Deepak Kumar had done on bacteria, we updated our model system 4 years ago. I presented this after some of the data that we had collected, and we wanted to start looking beyond this piece right here, beyond the ability of amyloid to stick to the HSV1 and agglutinate it. What other components and what other pathways could amyloid as an antimicrobial peptide be involved in, knowing that other antimicrobial peptides like LL37 are capable of many different functions beyond just agglutinating a pathogen. And at first, I'm going to go through each one of these pieces in the data that we've collected since then, and the story that we're building in terms of what amyloid and now tau can actually do. So in terms of opsonization, the ability of amyloid to kind of promote the capture and destruction of cells that may be infected, we simply looked at a culture, both A beta overexpressing and naive, and asked the question, where's the amyloid in relation to the HSV1? And looking at co-localization, we very clearly found that when the infection is present, the amyloid preferentially is binding to the cells that are infected. It's targeting the cells. Now, we have yet to figure out exactly what it's targeting. It may be certain signal cell receptors that are put on the surface when it is infected, or it may be remaining HSV1 glycoproteins upon the entrance into the cell. But again, we're seeing selective targeting of amyloid to cells that have been infected. And when we looked in our cell culture system of just adding synthetic or having an elevated expression of A beta 42 and examining how that affected the cytotoxicity, we found some very interesting results in that when we had no infection, just a normal cell hanging out, we had relatively low cytotoxicity as we would expect. In systems like the synthetic addition, we actually saw a drop in the cytotoxicity of the culture, suggesting that at certain concentrations, amyloid was actually protective for the cell. And then when we introduced the infection in combination with either the H442 overexpressing cell line or the addition of synthetic A beta, we saw this significant boost in the cytotoxicity, suggesting that amyloid is now working in conjunction with the cell and accelerating the death of these cells that may be infected beyond a system where you just have the infection. So now we have a model of the amyloid working to advance apoptosis or necrosis or panoptosis, which is a new one, to kill infected cells as a protective mechanism. A big one that is very interesting in terms of antimicrobial peptides is their influence on inflammation and downstream pathways of the innate immune system. They can actually act as signaling peptides and regulators of the inflammation response and can either accelerate it or depress it depending on what the microenvironment is and the infection. So we took our 3D culture system and compared the different conditions, trying to isolate how amyloid and the virus may be interacting with each other. We looked at AD versus control. So we have an amyloid overexpressing versus a naive to isolate the effects of A beta. We have control with and without HSV1 infection. We have AD infected versus control infected. So we're looking at the effects of amyloid in an environment with HSV1. And then finally, we have the AD infected versus the AD uninfected. So the effects of the viral infection in an amyloid overexpression environment. And this 480 hits of upregulated genes, these are specific only to conditions in which we have the infection, HSV1, and an elevated level of amyloid. They're not present when without the infection, and they're not present without the elevated level of amyloid, suggesting that this unique environment promotes um, a response. And when we examine some of these genes, unsurprisingly, we found things like the inflammatory response, TNF alpha signaling via NF kappa B, apoptosis, STAT5 signaling, uh, interferon gamma response. All of these are very important innate immune responses that are elevated when we have both infection and amyloid. And when we went a little bit deeper into upstream immune canonical pathways, we again found more pieces suggesting this connection to the innate immune system and the defense against an infection involving amyloid with innate immune inflammation, cell death, antiviral, and senescence pathways all being upregulated. Finally, we turned our eyes to tau. Uh, Obviously, we can't think about AD without thinking about both amyloid and tau. And while we know that there is this general progressive nature of amyloid to tau, tau to neuroinflammation, we wanted to see whether tau itself was actually an antimicrobial, and some of the responses and effects that we see with this hyperphosphorylation of tau could be more than a downstream effect of Alzheimer's disease. So first thing we tested, which was similar to what we did with the A beta, was can tau be protective? If we introduce a virus, HSV1, into the system, does the presence of tau reduce the amount of infection that we see? And yes, it does, but not the same way that amyloid does. When we ran the tests with amyloid, we were pre-mixing the amyloid and the tau, or the amyloid and the HSV1 before we were adding it to the culture. And what we were observing in that situation was that the amyloid had a chance to agglutinate and capture the HSV1 before introduction to the culture. So by the time that mixture was there was fewer viral particles free to actually contact the culture and infect. When we did that same sort of pre-incubation with tau, we didn't see an effect. And actually what we saw was an increase in infection. And what we suspect caused that was actually the viral particles being weighed down a little bit and being dragged faster down to the culture, resulting in increased infection. So what we did is we took a different approach and said, all right, tau is historically found inside the cell. So we would expect that if there is an antimicrobial effect, it would be intracellular rather than extracellular. So we pre-incubated the cultures themselves with the addition of synthetic tau, specifically the 2N4R GSK3 beta phosphorylated tau. So 2N4R is the longest form, isoform of the 6 that are present in humans. And we used a pre-phosphorylated tau, synthetic tau, with the GSK3 beta kinase. And when we pre-incubated the cultures before adding the virus, then we saw a protective effect. And this will be important and explained in a couple more slides, but it's interesting to keep that in the back of your mind. Not only did we see a reduction in the plaque count and the infected cell count, but we saw a reduction in the ratio of plaque to infected cells, suggesting that not only are we reducing the number of cells that are initiating replication of the virus, but we're also reducing the number of progression, the cells that actually get infected and then progress to a plaque and spread. That's also being reduced. And we saw that in the bottom here, where we saw a reduction in the plaque size. So it is helping to reduce that spread of HSV1 even after a successful infected cell. When we went to examine whether or not it could bind, much like how A beta 42 binds to the surface of HSV1, we found that, again, we had to look at what's happening inside the cell. And inside the cell, we're not seeing whole viral particles. The way HSV1 works is that it's an enveloped virus. It binds onto the surface of the cell and then merges with the cell surface to put the inner capsid into the cell. And that capsid is what travels to the cell nucleus and replicates the virus. So rather than looking at the whole virus, we isolated the capsids and examined whether we saw aggressive binding between capsids and tau, and we did. Tau was much more strongly bound to capsids versus the whole virus. And as a control, we took the VP2122 antibody, which helps targets a protein on the capsid, and we found that there was steric hindrance. As we increased the amount of this antibody, we saw less and less binding of tau, suggesting that we had our target correct, that it was the capsid and not the envelope. And then we tested the different isoforms. In the human brain, by the time you reach adulthood, it's primarily composed of the 2N3R and 2N4R isoforms. So we tested the two forms, a mixture, a 50-50 mixture of 3R and 4R, and then finally our 2 and 4R GSK3 beta phosphorylated. And while we did see some binding to the capsid of the other forms, the preferential form for binding was the 2 and 4 GSK3 beta phosphorylated tau. So now we've created this interesting connection of phosphorylated tau being our tau of choice for antimicrobial activities. And this is looping back into AD because much like the A beta 42, which we didn't really have a solid physiological purpose for, the phosphorylated tau is, again, something that is historically just connected to the development of these neurofibrillary tangles and not something that may serve a purpose. When we went and looked using EM, we did both a isolated synthetic experiment with just capsid and the phosphorylated p-tau, but we also went into our infected cultures looking to see if we could find associations between p-tau and virus in infected cells. And A and B show our synthetic experiments of just capsid and tau, and you can see basically two different forms of aggregation of tau. In A, you can see this irregular agglutination, this kind of clumpiness that is connecting these different capsid particles. And in B, you can see the fibrils. So this would just be a single fibril of aggregated tau going from one capsid to another. In C, what you're looking at is actually the nucleus of a cell. And the capsid of the virus is replicated inside the nucleus before it is exported out, enveloped, and then released from the cell. And interestingly enough, the immunogold particles that you see there are targeting phosphorylated tau. And we see that connection, that association between phosphorylated tau on the surface of these capsid particles and its location in the nucleus. So we have another key piece indicating that, yes, what we've seen in the molecular, the simple sterile experiments without cells also applies to our culture systems. So continuing on with our culture examination of tau, we kind of flipped to the other side. Rather than looking at how tau may interact with the virus, we wanted to see what the presence of virus does to phospho-tau in the cultures. So we have these 3D cultures. We infected them with HSV1, waited 24 hours to 48 hours, and then examined what's happening with p-tau. And as anticipated, we see an increase in p-tau, both in dystrophic neurites uh, and the cell bodies that have a positive p-tau signal. We also wanted to look at where this p-tau is going, not just that we see it overexpressed, but are we seeing things that would indicate aggregation, a movement towards neurofibrillary tangles? And we saw some of that, but it was also a little bit unexpected. What we saw here was an increase in the insoluble to soluble ratio of total tau and the insoluble to soluble ratio of p-tau in cell lysates. So that was supportive of this idea that we're getting a shift towards insoluble tau and potentially on our route towards neurofibrillary tangles. But what we also saw, which was a little bit unexpected, was a rise in the p-tau to total tau ratio in the media and a drop in the soluble p-tau to total tau ratio in cell lysates. And what this suggested was potentially that soluble p-tau is actually being released from the neurons out into the media space. And we wanted to know why. We were up to this point working in the assumption that everything's happening inside the cell. It's an intracellular innate immune peptide that's protecting against infection. What does it need to be doing outside the cell if amyloid is out there to kind of protect from the infection? And so that's where some of our unexpected results started to appear and modified our model. And what we started to see is that this soluble tau release was potentially another component of this innate immune protection pathway. And where we saw this was in the movement of p-tau from infected cells to uninfected cells. So in this experiment, we have a microfluidic device where we've separated the different cultures. They're all the same culture in each channel, but we're able to infect on one side and control the movement of the HSV1 from one side of the device to the other. It has to pass through the microfluidic channels, so it is using primarily the retrograde axonal transport rather than just diffusely floating through the cells. And the advantage that this gave us is we could create definitive regions in our image capture of red being infected neurons, yellow being neurons that were in close proximity to infected neurons, and then green being uninfected neurons that were nowhere near this infection line that was passing over the culture. And what we saw, interestingly, was that the uninfected cells that were close to the infected cells actually had a significant increase in the level of p-tau, and that the ratios were correlated. So if we had more HSV positive cells surrounding this uninfected cell, we had more p-tau intensity in this cell. And this suggested that the p-tau that was being released was actually being taken up by adjacent cells, almost in preparation for this impending viral spread. So we needed to try to separate this effect and really narrow in on, is this how it works? Are we getting a release of this tau and almost like a cytokine or another signaling compound is this spreading from cell to cell. So using inserts with a zero point 0.1 micron filter, we added HSV1 and azide tagged amino acids to the upper culture, and we grew those out. And then we moved this culture insert in above a naive 3D RenVM culture. And the idea behind this experiment was that with the azide tagged amino acids, now everything in that 3D culture we could see. And any protein that it produced, we could see using the confocal microscope because we could illuminate the azide tagged amino acids. And with the HSV1, we were now stimulating that culture to act as the infected line. But the pores on the filter were too small for the HSV1 to pass through, but they were big enough for something like phospho-tau to pass through. And what we saw when we looked at that bottom culture was that we had positive azide signal that had been taken up inside the naive, uninfected RenVM culture in the bottom, and that there was a very high correlation, almost 80%, between the p-tau signal and the azide signal. And that when we compared these azide positive cells to azide negative cells, they were significantly higher in this p-tau intensity. So now this really helps establish this concept that the infected cultures are releasing this p-tau as a protective mechanism for surrounding cells to uptake, preemptively boost their phospho-tau presence, and prepare themselves for a local infection. Another piece that we were unexpected for was this idea of amyloid impacting autophagy. Now, autophagy is important because it's one of the cell's primary defenses against HSV1 infection, and for that matter, many different intracellular microbial infections. What autophagy allows the cells to do is that they can ubiquinate the pathogen or microbe that comes in, and then through the use of various complexes and selective receptors, they aggregate the ubiquinated pathogen into autophagosomes, fuse with lysosomes, and then degrade this target. But much like many pathogens that have evolved along with us, HSV1 has developed counters against this. The ICP0 and ICP34.5 are both viral proteins that are meant to interrupt this process. So in this back and forth war, it's a balance. And usually, if you have that infection, the virus has won out on that balance. But there was a paper that came out maybe 10, 13 years ago that showed amyloid beta hanging out in these LC3 positive vacuoles. But that's as far as they took it, just showing that there was LC3. And at that time, the premise was, well, amyloid 42, much like a microbial pathogen, may be getting broken down and released through this autophagosome process. But we wanted to say, well, what if amyloid is actually part of this pathway? And when we started looking into these different components of the autophagic pathway, we found that amyloid does play a impactful role. When we remove amyloid in amyloid knockout lines, or we have a elevated level of amyloid through A beta overexpression lines in our 3D cultures, we see this shift, this decrease or increase in the SARs, Bestlin-1, which is an important complex that HSV1 goes after, and LC3-2, 2, which we showed is important in making those autophagosomes. So now looking at the amyloid beta manipulating this increase and decrease, what happens when we add this infection? We know the infection will suppress autophagy, but if we have amyloid, a lot of it or none of it, then what happens to the autophagic process? And without amyloid, the autophagy is more suppressed. And we see that both in this decrease in Bestlin-1 and P62. And alternatively, when we look at this conversion of LC31 to LC32, in the overexpressing cell lines of amyloid, we actually see an increase in LC32. So now in the presence of virus, without amyloid, we're seeing a increase or a decrease in autophagy. And with amyloid overexpression, we're seeing an increase in this LC3-1-2 process, suggesting an amplification of autophagy. And when we turn to our immunofluorescence, we used a drug called baflamycin. And what baflamycin does is it essentially stops the autophagy process right at the end before it merges with the lysosomes. So anything that's put into an autophagosome now kind of gets backed up and stuck. And it allows us a better chance to see what's going on before all those components are broken down in a lysosome. So again, when we have this comparison with APP overexpression, we see a buildup of these P62 A beta 42 positive vacuoles inside these cells in the presence of HSV1. And when we look at LC3B and this conversion of LC3-1 to 2, we see an even stronger increase when we have more amyloid. So we've now looked at not just what amyloid and tau can do in terms of the immediate effect of a pathogen that is in the area or has initially infected a cell, but also shown that these innate immune peptides or host defense proteins, as Rudy and I have decided we're going to push as a way to account for tau being unique in its size. We've shown that they affect multiple pathways and multiple steps in this infection process, all the way from entering into the area to breaking down or isolating cells that are infected. And the deeper we delve into this, the more complex and interesting the story becomes. And where we are now is we see all these different types of microbes, from bacteria to parasites, like toxoplasmosis if you have a cat. And each of these pathogens has unique properties. They're not all the same. They don't all have the same surface proteins. They don't all travel through the body the same way. Some of them have preferential targets for neuronal cell types. Some of them like to hang out in endothelial cells. Glial cells are a favorite target for some parasites. And in incorporating not only the cell types, but the areas of the brain where these pathogens may be hanging out. So there's quite a diversity in what the central nervous system is facing in terms of pathogens. And we think that the piece that we've described so far is actually just a tiny portion of what the innate immune system inside the brain is composed of. There are other peptides and proteins that may be historically associated with disease that are starting to look more and more like uh, antimicrobial peptides, host defense proteins. And not only that, but we're finding that even modifications like the phosphorylation in tau is capable of altering the selectivity of tau and its antimicrobial properties. So there's a whole ecosystem that seems to be present inside the brain that we have yet to decipher. And the more we piece this apart, the better we're going to be able to understand how we progress from an infection. And it could be a whole host of different infections. And it's targeting into the brain, it's destruction of cells, and this progress towards Alzheimer's disease. And hopefully, by uncovering these pieces, we can also find early intervention targets, either through targeting these pathogens themselves or manipulation of the innate immune defense, amyloid and tau, or whatever other uh, proteins we find to help bolster the defense before we see the neuronal loss. Um, Much like we've been discussed previously with getting in there and modifying amyloid presence early on. So it's an interesting story that we're excited to chase down. We're honored that Rob started this for us and came up with this really out of the box thinking. And Doctors Deepak Kumar and Nanda Shanugam have been working with me and helping push these ideas forward. And with the guidance of Dr. Rudy Tanzi, we've made some real strides forward and continue to uncover this really fascinating new area of innate immunity inside the brain.
Okay, William, thank you very much for that excellent presentation. Very interesting. Um, so we're going to open it up for questions and discussion. If you do have questions, you can put them in the chat. You can also raise your hand.
Hey, Gene, I can start. Hey,
Okay.
nice job, Will. And I guess the question is, there are groups that are trying to do antiviral clinical trials on patients, Alzheimer patients. And the question is, if you do an antiviral trial, are you going to help a patient in terms of treatment, or is it going to only be useful for early prevention? What's your view on that?
That's a good question because while the obvious result is we have a antiviral, if they have a vaccine or they're treating with acyclovir, we're reducing the presence of virus. So in that regard, we are taking this innate immune response and wiping away what would trigger it. So we should theoretically see a downstream decrease of risk. But the caveat to that is that we're dealing with the innate immune system, not the adaptive immune system. So while it's capable of finding these pathogens, it's capable of finding a lot of different things. And we have to recognize that anything from a bacterium to possibly even pollen to a traumatic brain injury is all capable of initiating an innate immune response. So while the decrease of one piece using antivirals will and should help the community in terms of preventing the progression, and or if someone has something like HSV1 that is a persistent infection and has reoccurring episodes, it should help suppress that. there are going to be other pathways, and that's evident in the Taiwanese study that did a retrospective look at people treated with acyclovir and whether they had active infections of HSV1 or not. And it helped. It reduced the risk of Alzheimer's disease, but what it didn't do was eliminate the risk. What it was effective at doing was bringing the risk back down to the baseline of people that were not infected with HSV1.
Okay, next question. I think it's Stu. You have your hand up,
Oh,
please.
yeah. Thank you very much. It's a nice talk. So I have two questions. First is that clinical implication. You know that A-beta, anti-A-beta, anti-tau immunotherapy has been already studied. So based on your observation, do you expect this is going to be patient more vulnerable to the certain type of viruses? I think that's the first question. And second question is alternative hypothesis mechanistic. So there's a lot of studies done by internal tau protein and phospho-tau, whether they interact with the double-stranded RNA and double-stranded DNA inside. So I'm wondering whether those interactions with tau, with these viral DNAs, herpes virus in this case, can be the potentially protective mechanism in addition to interaction with the capsid protein.
Yes. So the first question about these anti-A beta, anti-tau drug treatments. That is a two-part stepping stone. And the first is that many of the drugs and treatments, like the anti-monoclonal antibodies against A beta 42, are very effective at wiping out amyloid in the brain. There's some beautiful images of it just being gone. And while that is great, from our point of view, you've stripped a piece of the immune system out of the brain, and that runs the risk of getting you exposed to a potential future infection.
So
Well,
that's actually before going into the second question, like so if that was the case based on your data sets, like these patients with anti-tau antibody therapies may more vulnerable to HSV infections, right, in the brain, right? Am I correct? That's actually what your data suggests.
Yeah, but it's also going to be that they're gonna need to be to see the effect of that. You're going to have them continuously on these drugs that are going to continually wipe the presence of either amyloid or tau from the system, and that's going to create the vulnerability. From our perspective, what would be more productive would be regulation. We don't want to eliminate it. We want to reduce it, control it, much like we control statins in heart disease. If we keep it from getting to a giant level, then we can help manage that innate immune response. And in connection to your other question about tau and this double-stranded DNA, the simple answer to that is absolutely. There are multiple studies out there that have examined the C-gas sting detection system that captures the foreign DNA or DNA that at least perceives as pathogenic and activates the immune response. And interestingly, both the other studies and some of our own studies, what I didn't show you in the autophagy work was that C-gas sting is also changed in the shift of amyloid and the change in autophagy, macroautophagy. So that fits in with this idea that tau may be another component against invasive DNA or perceived invasive DNA and another route for innate immune response.
Okay, thank you. We still have about 8 minutes. Are there any other questions?
Well, you know, there are lots of groups planning active vaccination against A beta, and now there's talk of active vaccination against tau, which I have a lot of concerns about because everyone has a different immune response to an active vaccination. And so you might have a person with a very strong antibody response, and you're wiping out A beta monomer if the A beta vaccine, for example, only doesn't just see oligomer and fiber, but it sees monomer. You might wipe out monomer, which is a host defense peptide. You might hit p-tau, which now you've shown is a host defense protein. So can we get any sense of, how can we monitor it in an active vaccine trial, whether the immune response is too high? I mean, would you worry about wiping out too much p-tau with an active vaccine?
I'd be terrified. Um,
Glad you said it.
Yeah, no, that prospect's absolutely terrifying. And I think what baffles me about that thought process is this idea that amyloid, A beta 42 and oligomerized amyloid and phospho-tau aren't serving a purpose. The sequence for amyloid is highly conserved, going back hundreds of millions of years. And we're going to rummage around in there and strip it out and say, well, it's bad. We don't know what it does, but we know it's bad. I here just touch on the innate immune functions of amyloid. There are plenty of other groups that are studying other physiological functions of amyloid, including helping maintain the
of A beta, not amyloid, but
well, yeah, of A beta, including helping maintain the vasculature structure in the brain, which I suspect is a little bit of why we see the ARIA brain bleeds when we do the monoclonal treatment. I think we're stripping away some of what's helping glue everything together. So, yeah, and I think that you're going to have a dual problem with this vaccination system because not only are we making these people essentially immunocompromised, so we're going to have to be monitoring for potential infections, but you've also short wired this innate immune response that we are shown, with the autophagy. Again, I go back to the autophagy. That's not just a system that's used for pathogens. That's a system that's used for cell health and maintenance in the brain. And if we're stripping out a component, is it going to stop it? No, it's going to continue, but we're going to modify it. And who knows what the downstream effects of modifying that in terms of brain health are going to be.
Yeah. And remember the base inhibitors that really knocked out A beta production made people cognitively worse. I think that's also because A beta plays a role in regulating glutamatergic transmission, so that when you have glutamatergic excitation, A beta is released together with zinc, and those oligomers then induce the reinternalization of NMDA receptors to turn off the signaling. So, I mean, there's lots of roles for A beta, and it's just kind of scary that people still look at it as a bad guy and not listening to what, Rob originally conceived of. The amyloid hypothesis really began with George Glenner in 1984 and 85, which is 40 years ago. And even Glenner, when I met him back at, when I was cloning the gene, said he thought that A beta was not just junk. He thought it, he said like other amyloidoses, the protein that turns into amyloid has a normal role.
Yeah.
There's no amyloidosis where the protein that turns into amyloid, whether it's transthyretin and cardiac amyloid or IgG and splenic amyloid, doesn't have a role. But people forget this stuff.
A last general question. Great talk. Well, but I have a general question concerning other clinical trials in Alzheimer's disease. Do you think that can these findings influence the patient stratification of patients included in clinical trials that are maybe clinical trials of the A beta antibodies that are not necessarily linked with the antimicrobial theory of that you presented, but like are targeting other mechanisms in Alzheimer's disease. But do you think that the infection status is important in general application of AD drugs?
Absolutely. I think that is a clinical status that has been ignored in terms of preparing the patient population. And, an easy example of that is sex. So we know that females are way more likely to develop AD. It's 60, two-thirds higher. For every like 3 patients, 2 of them are going to be women. And what is not thought about in that process is that women are actually also more likely to get and maintain HSV-1 infection. They're more susceptible to it. And part of that is in the significant differences between the immune response and systems between men and women. Women are more likely to develop autoimmune disorders. They tend to have an immune system that maybe is not as initially aggressive as men's, but is more likely to persist and maintain in terms of an immune response. So when we ignore an infection status, people who have had, as Dew pointed out in chat, people who've had chickenpox and shingles, people who have different types of persistent infections, people who come from different regions that may or may not have the presence of, a higher presence of pathogens. One of them that Rudy and I have talked about briefly is helminths. They're a parasite that are way more present in Africa. But the interesting piece of it is that they are immunosuppressive. So when you're infected with them, your immune system is actually suppressed. And that reduction in the inflammation and reduction in the response, maybe that plays a role in why there seems to be less incidence of Alzheimer's in Africa. It's a maybe when we start connecting all these pieces. So I think that people need to start paying attention to what these infections are, what they've been exposed to, not only as a persistent infection, but as historical. Did you have meningitis? Did you have encephalitis? Did you have tetanus? What other things have you contracted from your
Interesting
childhood?
after the COVID epidemic.
COVID is going to be a big one. There are already studies showing that COVID, the spike protein, is sufficient to activate the amyloid response and progress plaque development. And I think that as the generations that got hit the hardest with it reach elderly age, you're going to see an acceleration of AD prevalence, particularly those that may be faced with long COVID, where there's a persistent pool.
Yeah, but luckily, COVID doesn't replicate well in the brain because of the lack of the receptor. So it's maybe not quite as bad as others.
But unlucky, it can hang out in the gut tissue and release spike protein into the blood, which can reach the brain.
That's true. Yeah, that's true. That would be recognized as a target for AMPs. Yeah.
Yep.
Okay. William, thank you so much for coming and sharing updates and for your talk. Thank you, everybody, for your attendance, and we'll see you in a couple weeks on a Friday. Have a great week.